Thank you very much for coming along. We're excited to have a fantastic lineup of speakers today, and they are from the College of Science and Engineering. The speakers we have today will be talking about their research opportunities, and for the students, you might want to engage with the professors to be able to take advantage of those opportunities. Some of these professors have supported students for a very long time, like Professor Walcott and Professor Scorban, and some professors are brand new, fresh, looking to connect with students. They just arrived. They're energetic and passionate about their work, like Professor Yao Ezunke. So Professor Yao Ezunke is going to be our first presenter, and he will tell us all about the research opportunities in his lab or program, and people will be able to contact him from there. But please, Professor Yao, the floor is yours.
Thank you very much, Professor Folaren. Sorry, I did not put any PowerPoint together. I'm just gonna talk. It's a great honor to be part of this panel. For those who have not met me yet, my name is Yao Ezunpe. I graduated from San Jose State Aerospace Engineering Department. I went on to receive my PhD from the University of California, San Diego in engineering science in aerospace. So my research is basically in a thermofluid applying a computational mathematics. What I mean by that is I do theoretical and computational fluid mechanics. So I employ technique in applying a computational mathematics to investigate basically fundamental problem in transfer phenomena that take place in domain bound by highly irregular surfaces, complex multi-phase fluid structure interaction, biomedical fluid, and microgravity sciences. So before joining San Jose State, I was a lecturer at San Diego State Mechanical Engineering Department, UC San Diego Mechanical and Aerospace Department. So if you have any question, I'm open. I'm still building my lab now, so we'll have plenty of opportunity to work with a student. I started my research as undergraduate from San Jose State, where I was working with one professor in Texas A&M in the aerospace department through LSAMP program. So I'm grateful that we still have those programs, and we will be taking advantage and make sure we get people that will be successful. So if you have any question, I'm open.
Moving to our next presenter, Professor Walcott. He has supported a ton of students in his lab. He's received at least $1.5 million in funding at SJSU, which is a good number compared to a lot of professors. And he's worked on some of the cutting edge work I'll let him talk about it. Professor Walcott, please, welcome.
Very good. Thank you for the introduction, and thank you for the organizers for putting this together. Yeah, I'm in the Department of Chemistry. I'm currently an assistant professor, but hopefully within a couple of months I'll get tenure and then be an associate professor. The name of my laboratory is the Surface Science Center Lab. I am the manager of the laboratory, and we focus on the surface structure of bulk in nanoscale materials. And nanotech was big maybe about 20 years ago and has faded as other things like CRISPR and other technologies have come into play. But nanotech is still there, and it's definitely important for us to consider the implications and how nanoscale materials can be used in biological applications. Up on the screen right now is the sun-drop diamond, and this is the world's largest nitrogen-doped diamond. And what this is giving us is a kind of a picture into the type of material science that we do. Even though we investigate nanoscale diamond, the nitrogen atoms that are inside of the diamond, we then use to make a special atomic defect called the nitrogen vacancy center. Also, the surface properties of this large stone and its 25 nanometer counterpart are almost indistinguishable from one another. And so our job is to take this little tiny nanoprobe and begin to control its surface properties with a high level of specificity so that we can take it into cells and tissues and into other materials so that it can be used as a quantum sensor. The whole idea of it being a quantum sensor is based on this nitrogen vacancy center. So imagine diamond is all carbon, but you can take out a carbon atom and substitute it in for nitrogen. And then if an adjacent carbon is then knocked out with a high energy beam of helium atoms or hydrogen atoms or electrons, you play atomic billiards and you knock it out and you generate what's known as a vacancy. And so when this nitrogen atom and this vacancy are right next to each other, it creates this unique platform to be able to sense what's going on around it. And so this is a magnetic and electric field sensor. And we can use the term quantum bit or a qubit because we can understand how the system behaves using quantum mechanics. This is the fluorescence of the system over on the right-hand side. There's an NV0 and NV minus charge state, so it kind of switches back and forth. It has a couple of different flavors. And then the electronic structure, how it behaves, is depicted over here in the bottom left. And the way that you can view it is down here at the bottom left, there's a 0, +1, and -1 state for the electrons themselves. So you can imagine that it's similar to what we would have in a regular computer, having zeros and ones. But now we can have this extra space in order to encode information and to sense what's going around it. Imagine that the single electron that's inside of the NV center is similar to the smallest compass needle in the world, and it's highly sensitive. So if somebody is moving a fridge magnet 6 miles away, the NV center, if you're watching it, will begin to wiggle when that little fridge magnet is moved. Why is this important? Well, we can end up putting the diamond that is the host of the NV centers into cells and in tissues. And so it's a powerful platform to do quantum sensing. Here are some HeLa cells that have been stained with nanoscale diamond. Below it on the bottom left is room temperature NMR spectroscopy. Many of you who might have taken organic chemistry remember NMR. Well, in the NV center community, you have optically detected magnetic resonance. So you can do this at room temperature. And on the right-hand side is some work that I did with some colleagues back east, where you can use the NV center for electric field sensing, which could be important for pH sensing or for measuring action potentials, basically watching neurons fire in real time. Now, the idea, though, is to be able to have new tools, new fun things to do with this diamond material. For us, we don't just do the work here at SJSU. We also go up to the Stanford Synchrotron Radiation Light Source, which is only 25 minutes away. And we also go to the Molecular Foundry, which is at Lawrence Berkeley National Lab. And so we get the idea that we can use these special tools to look at diamond and get very, very specific information about the system. This is called the RIX map, which tells us about the ground state and excited state electronic structure of material, which tells us about the molecular groups. Is it an amine or an alcohol or a carboxylic acid that's on the surface of the diamond? These cool tools allow us to have the ability to understand this information. And so when we're at Stanford, we get to use a very special sensor. It's called the transition edge sensor, which is a superconducting X-ray sensor. And what it is basically is it's the world's most sensitive thermometer, and it's held at 60 millikelvin, so only 60 millikelvin above absolute zero. So we are talking about something that is super, super cold. And here's myself and two of my students, Tayana Supreme and Daniel Labunsky. Over on the right-hand side is where you can see the transition ed sensor. It kind of looks like R2-D2 or something along those lines, but it's basically a camera. An amazing camera with a high level of sensitivity and has almost no background signal when it's detecting these x-rays that come off. Now, before you get into PChem, you might see something that looks like this. This is a molecular orbital diagram. And basically this illustrates what's going on with our system when we hit it with x-rays and place electrons into unoccupied electronic states. So you can imagine it's almost like filling an electron into some sort of quantum state that's higher up in the building. It starts off on the first floor and ends up on the third, fourth, and fifth floor, and is illustrated right there. I've got 3 different thrusts in my research lab. There's openings in every single one of the research thrusts. It's very exciting and interdisciplinary research group. We work in groups of 3 to 5 people. There's Team Amine, Team SiO2 Plasmonics, and Team Boron. Whether or not you're interested in physics, materials, chemistry, or even biology, we've probably got a place for you. We've been very successful into getting former group members into PhD programs. And basically, we've got the available funding to support students during their research careers. And if you'd like to reach out to me, you're more than welcome to. Thank you so much.
Thank you very much for that presentation. As mentioned, Professor Walcott has a lot of experience, and you can see where his students go. What you saw from his presentation, you may not understand now, but if you spend some time in the lab, reach out to him, you'll gain some insight, and you start speaking like that too. And people be like, wow, you know.
Lots of jargon. Sorry, everybody.
So please reach out to him, and if you want, his LinkedIn and email are in the chat. Our next presenter is from physics. So we're going from chemistry to physics. Professor Kasahun Betri, please go ahead and take the floor.
My name is Kasahun Betri. I'm in the Department of Physics and Astronomy. My area in physics is called high energy theory, which is an area where we try to study the most universal and the most basic or the most fundamental building blocks of nature. How do we do that? We do that by constructing models, and we try to explain something by appealing to something that's more basic or more fundamental, conceptually speaking. And I focus on space, so that means we're trying to understand what space is made of, what its structure is. Now, immediately you may see a dilemma. Space usually in any physical theory, space is a starting point. It's something that we take for granted. We start by assuming there is space and there's stuff in the space, and then we explain how stuff behaves by appealing to its relation to space. But if you're trying to explain space itself, how do you do that? What can be more fundamental or more basic than space itself? So it turns out, well, there are things that are more basic and more fundamental than space, and these are relationships between constituents and probability. So what I work on is to try and build space up, conceptually speaking, by using kind of relationships, a type of representing these relationships, which are graphs, and by appealing to the probability, specifically statistical physics. And actually, what I said is not just motivated from philosophy, but also from actual high particle physics. Right now, there are two different theoretical foundations for that modern physics is resting on, which is, one is quantum field theory that explains the very small units, and then the other one's general relativity. It's a classical theory which explains nature at the largest scale, and things that have extremely large energy or occur at very large scales. But these two theories have not been successfully merged into one in such a way that has been experimentally verified. But there are good reasons to think a unification of the two is needed. A sort of quantum gravity theory is needed because there are things that are both extremely small and also extremely heavy or energetic, like black holes. And to understand really what happens inside a black hole or other singularities such as the Big Bang, you need a quantum gravity theory. And the traditional way that people try to find a quantum gravity theory is by starting with a classical theory, and like Einstein's general relativity is a classical theory, and then you apply some a procedure called quantization, and you try to find a quantum system. That has been the main effort that has undergone the past several decades. Recently, though, people are also beginning to think maybe we can start with something that's truly quantum and try to see if if something that appears to be a space-time can emerge as an average large-scale property. That's called emergence, and that's what I work on. And the idea is you start with truly quantum building blocks that made no reference to space. And so how do you refer to things? Just through relationships, intrinsically, self-referencing. And then see if you can, as a large scale average, if you can get something that looks like space. And kind of going into the detail, the theory is built on graphs. Graphs are things that are made up of nodes and connections between the nodes, which are edges. And so here's an example of a graph where we have 6 nodes. And in this case, like node 1 and 6 are connected. So there's an edge between them, but node 6 and 3 are not connected. So there is no edge between them. And so the idea is you have some very large n number of nodes, and you take all possible relationships between them. That gives you all possible different graphs. And that constitutes kind of the possible configurations of your system. And each configuration or each graph is assigned some energy given by a Hamiltonian. And that allows you to do probability because you have a very large number of these. You kind of look at average or macroscopic averages of these. In other words, you apply basically statistical physics. And the idea is you start with something that's truly random, right? Like on the left, but then there's some kind of phase transition similar to the way water freezes and locks into a regular structure. And what you get there is something that looks like discretized space. And so the research involves graph theory, statistical methods, and also numerical Monte Carlo simulations. You don't need any of these skills. You'll pick them up as you go. And no prior is needed. The only skill that would be good to have coming into this is being able to program in any language. And the rest, you will learn as you go. And you do not have to be a physics major. You can be, engineering or math or other majors. And I'm looking to hire 2 undergraduate students and 1 master's student. The research will be for credit for units during the school year, and you'll be paid during the summer. And so if you're interested in hearing more about my research and participating in this, please contact me. This is my email. It's also in the chat. Thank you so much.
Fantastic work. If you're interested, please contact Professor Vetri. Next up, we have Professor Elizabeth Scorban from the Biological Sciences. So please go ahead.
My name is Elizabeth Scorban, and I'm a microbiology professor in the Department of Biological Sciences. And we are recruiting students to start over winter break for the spring semester and also for summer. So I'm going to just give you sort of an overview or summary of the main project in my research lab, which is to engineer this bacterium shown here, Methylorubamixtorquins, to recycle rare earth metals back out of electronic waste. And so if you haven't really thought about rare earth metals before, they're shown here on this periodic table, highlighted in red. It consists of 15 lanthanide elements, along with 2 chemically similar elements, scandium and yttrium. And why we all care about rare earths is they basically make our technologies run. So they're essential components of our computers, of our smartphones. They're important for our green energy technologies like our wind turbines and our hybrid car batteries, medical diagnostics, so very, very important. But the issue is that mining for these rare earth metals is very environmentally destructive. Radioactive byproducts get released into the groundwater, which kills off a lot of biological diversity and is really toxic, of course, to the people living in around those areas. And so as a result, we mostly import our rare earth metals from other countries. China is the largest provider. And so we thought we would engineer this bacteria that can acquire rare earth metals naturally to instead have an environmentally friendly, cost-effective biological platform to recycle rare earths back out of things we just discard, like our discarded electronic waste. And so in order to be able to do this, we have to know a lot about how the bacteria acquire these insoluble rare earth metals, bring them into the cell, and what they do with them once they're there. And then we can engineer the cells to do what they do naturally better. So we spend a lot of time figuring out how rare earths are acquired and transported. And the cell is smart. It only takes up what it needs, because otherwise it's a waste of energy. But we don't want it to stop taking up rare earths when it's had enough. So we engineer it to bring in more and more. And then what's very interesting is we found that when the cell has excess rare earth metals, it stores them complex to polyphosphate in the cell. And they form these little granules that you can see here in this microscopic image where these polyphosphate granules are stained. So essentially the bacterium is concentrating the rare earths that it's acquired from electronic waste, and it's purifying them. So that's the only other metal that's found in this polyphosphate granule. And then we can extract these granules back out of the cell and recover the rare earths, and they can go back on the market. And that's done in collaboration with the Martinez-Gomez group at UC Berkeley. And we also partner with Lawrence Livermore National Labs as well. And so in addition to just trying to figure out how the cell does what it does naturally and engineer it to do it better, we have a couple of hurdles that we have to overcome. And the first is that electronic waste is very toxic. It contains all sorts of nasty heavy metals like mercury, cadmium, lead. So not only toxic to us, but toxic to the bacteria as well. So what we've done is we've taken smartphones as an analog of electronic waste, pulverize them by blending them in our laboratory, and then we can add that to our growth medium, which is why these plates look black here, because they contain pulverized cell phone. And the strain that we start with, our wild type strain, can't grow, but you can see that we've isolated strains that are resistant to the electronic waste. So we're trying to figure out what mutations have occurred in the DNA that allows this new ability. How are these mutations leading to this resistance? And then finally, one thing I didn't tell you, but the bacterium that we work with can't take up all 17 rare earth metals. It only uses 5. And so we've engineered new strains of these bacteria that can use rare earths that it normally doesn't use, like gadolinium, which is important for MRIs, for diagnostic tools in medicine, europium, which is a phosphor in your cell phone screens. And so we're broadening the scope of rare earths that our bacteria can recover. And so with that, I'm happy to take any questions, and I don't use LinkedIn, so please email me if you are interested in working in my lab.
Thank you very much. Our next speaker is Patrick Journey. Professor Patrick Journey, please go ahead.
All right. Thank you, Dr. Eros. Okay, first of all, thank you for inviting me to give a talk today, Dr. Eros, and thanks for organizing. This is fantastic. So can you all see my screen? All right, we have, okay, good. All right. So in our lab, so as Dr. Earl said, I'm relatively new. We're beginning our 5th semester here at SJSU. And in our lab, we use microfluidics and other tools really for 3 different purposes. All of them revolve around the human circulatory system. So we have expertise in nanoparticle drug delivery. And I've given an hour talk that Dr. Eros has listened to, so I won't talk about that at all today. And Dr. Walcott left, but he's also suffered through that talk in the past as well. But the other 2 areas I will talk about today, which are synthetic vascular grafts, as well as the study of the biochemistry of thrombosis and hemostasis. And I'll talk about what both of those mean and how we address them here in a second. So we're really interested in the balance between hemostasis and thrombosis. Thrombosis, if you look in this picture here, is really a pathological response of a totally normal and healthy biochemical mechanism that we have in our body. So throughout the day, you have hundreds to thousands of little micro events, micro cuts in your vasculature that need to be repaired. And the sort of like vehicles that repair those cuts are platelets in the body. Those are about 1 micron little cells that float around in your vasculature. And they sort of initiate the process of closing that wound, that vascular wound, and then initiating a healthy regenerative response. So, but when it runs away, it turns into thrombosis and heart attacks and that type of thing. So in our lab, we combine some material science, some fluid mechanics, and some biology of the cells that make up the lining of all of our vasculature, which is known as, they're known as endothelial cells, into our sort of engineering expertise to see if we can engineer some of these systems to be used in the clinic to treat some of these cardiovascular diseases. So I won't spend too much time on this slide, it's very busy, but we do this by really attacking understanding 3 interfaces. Those interfaces are between the biomaterial here. So I have like a picture of what would be maybe a vascular graft or a healthy vessel. You have this sort of basal layer, which would be the biomaterial. Then you have these endothelial cells attached to that material, and then you have blood flowing on the luminal surface of those cells. So we're really interested in the surface, the interface between the cells and the biomaterial, between the cells and the blood, and between the biomaterial and the blood. And there's a lot of chemistry and a lot of mechanics that go into understanding these surfaces, but that's basically what we do in our lab. So one example of what we do is this is what is called the coagulation cascade. And there will not be a quiz on this figure. Frankly, I couldn't recite it from memory, but it's a series of sort of related biochemical enzymatic reactions that ultimately lead to clotting and this healthy process of hemostasis on one hand that I discussed, or thrombosis on the other hand. And so one sort of thing that we have developed in the lab is a microfluidic chip to simulate that hemostatic process or a vessel injury, and then to understand the process of hemostasis. So we manufacture these microfluidic chips in the microprocess engineering laboratory here at San Jose State. And it's basically a microfluidic device where you input blood here, and we have these pillars which sort of simulate a rupture of your vessel. And then we allow the blood to flow through, and we see how the body repairs that injury site. And we've done this. And so here's a video of blood flowing from the bottom to the top of this frame. And you can see as platelets activate and you start to develop this clot at the vessel injury site, you'll see eventually we'll get a full hemostatic plug there. So then you've restored normal blood flow in the vessel, and the injury site can begin repairing. Now it's a multi-step process, and the nice thing about microfluidics is we have control over what's in all of these channels. So we can coat this channel with different proteins that appear in the body and see how that affects our hemostatic process. Now, one thing that we do in the lab is simulations of these, so numerical simulations, computational fluid dynamics, to understand some of the response of our platelets to specific flow fields. So this is work that an undergraduate who has since graduated did in the lab during the pandemic. She was able to do these simulations. Now she's at UCSF Berkeley in their master's program. And here's more uses for our chip. We can, flow and get different flow fields out. We can basically set the clots and then stain them for different adhesion molecules within the clot. And we can also do various clot mechanics. So you can see we've built a big clot here. This is really thrombosis more than hemostasis. And you can see the clot kind of responding to the flow in our chip. And so this is to say that I don't only take biomedical engineering students in my lab. This is work that Aldrich Estanilo, who was in the chemical engineering department, did. And we've developed a large 43 equation set of ordinary differential equations to describe this clotting process or the coagulation cascade. And we worked with our partners up at Oregon Health and Science University to do some experiments to sort of understand clotting on a reaction by reaction basis. And now we have a mathematical model so we can go in and parse out some of the responses that we see. So that's what's going on in the lab. So I'm taking students, feel free to email me. I don't know, I haven't watched the chat, but I have a LinkedIn. And also my email is patrick.journey@sjsu.edu if you're interested in microfluidics fabrication, any of the biomaterial stuff or the blood stuff. We have one student who is currently a McNair scholar working on some of the biomaterial stuff. So we can, I sort of wish she was here, but she's not, to Brittany Butt. So you can ask her what it's like to work in the lab. Hopefully it's positive, but I'll be separate from her review of the lab. And this is, so I think my time is up, but here's who we have working in the lab currently, as well as our collaborators. And this is an opportunity. It's not often that you get a chance to be the first, but I have recently started a Twitter for the Journey Lab, and we currently have 0 followers. So you have the chance to be the first follower. Go there at Journey Lab Twitter, and we're going to start publishing publicizing some of our fantastic work. So thank you.
All right. Fantastic work by Professor Journey. Please reach out if you're interested. Next up, we have Professor Scott Schaefer.
So my name is Dr. Schaefer. I'm a professor in the Biological Sciences Department. I have the Animal Ecology Lab. Most of my research and my graduate students study the ecology of seabirds primarily. My background is actually physiological ecology. I was a graduate student at UC Santa Cruz, but I'm also a CSU alum. I went to San Diego State and then went to Santa Cruz for my graduate work. So what I've shown here are just some of the graduate students in my lab, and you'll notice that most of them are holding a bird of some kind, and that's primarily because most of our work is field-based. And I just have one other slide to more or less show you the kind of work that we're doing. One of the primary objectives of the research in my lab is to understand the where animals are going in the environment, and our primary focus is on seabirds, so the marine environment. And we want to know where the animals are going and what kind of environmental features attract the animals to go to those places. So one of our primary modes to understand this is by deploying biologging devices. And so this is a rhinoceros auklet that you can see in the upper left. And we attach these GPS tags. They're just small GPS units that record at a high resolution. And we catch the birds at the breeding colonies. So there's a variety of islands we work at, both here locally and also abroad. And the idea is, again, to track where the animals go and then look at the environmental features. So in the figure on the right, you see this is San Francisco. This is the peninsula here, and right off of San Francisco, about 30 kilometers, is the Farallon Islands. And this is one of the locations we do some of our work. And these black lines represent the track lines of the animal as it's moving through the environment to its destination. And we can relate the animal locations to environmental features, oceanographic features, using remotely sensed data. So in this case, I'm showing sea surface temperature and the frequency of temperatures that the bird is most commonly associated or found in. And we can also collect diet samples. So the birds will come back with fish, and we can collect that fish either from the chicks or from the adults themselves. And it gives us some information about what the animal's doing, what environmental features are influencing where it's going, and what is the prey base that it's acquiring. And based on that, we can develop some ecological models that are predictive in nature to say, okay, if we see these kinds of oceanographic conditions, we would guess that the animals would be attracted to those features. And that has all kinds of implications for both species management and conservation as well. So my students and I do a number of projects that are similar to this. And then more recently, I'm collaborating with one of our microbiologists in the department, Dr. Kleber Overnay, and we've developed more of a metagenomics approach. So we've got several species like this Western gull, which we track. So we place a GPS tag on it, and we know that many of these birds will utilize habitats like landfills and garbage receptacles, but they also forage at sea. So they're not just targeting landfills, but we know based on the birds going to those locations that they pick up bacteria that could potentially be harmful to those individual species. And so what we do, in addition to tracking each individual, we collect swab samples of the mouth and the cloaca and the feet. And then some of my students are working in Dr. Overnay's lab to extract the bacteria. And then we do the sequencing and then a bioinformatics analysis to look at the different microbial diversity that we find on the birds and how that relates to the habitats that they're going to based on the GPS tracking and again, tying it to these environmental features. And so I've got a couple students working on some of that work at the moment. As far as undergraduate opportunities, I did just get an NSF award that I'm a co-PI on. And I'll probably bring in at least one undergraduate to do some analysis with diet samples, placing, IDing the prey samples, and then also placing them in an oxygen bomb calorimeter so we can determine the energy content. If you're interested in opportunities, let me know. I didn't post my website or my email, but you can find me on the biology website and just send me an email and we can set up a meeting and chat. Yeah, I'm happy to take questions. Thanks.
Thank you very much. Appreciate it. Next up, please. Let's hear from Professor Philip Heller.
Yay. Okay. Thank you for your patience. And thank you to my host for putting together this, this really good idea. It's really important in San Jose State that undergraduates be exposed to research as early as possible because it's such a great part of education. So what I study, my name is Phil Heller. I'm a professor in the computer science department. I do bioinformatics. And where do I do it? Well, if you look down deep enough into the ocean, or if you look up high enough into space, that's what I study. And one of my projects is called Coral Vision, which is where we study the dynamics of population change under climate change of coral reefs. So corals fringe a quarter of the world's coastlines, which is just an amazing amount of distance. And they support millions of species. They've been called the rainforests of the ocean. And why should we care? Well, they demonstrate just stunning beauty like this. And economically, they have a pretty big economic impact, but let's forget about the economics. Every time we breathe, we're breathing oxygen that was put in the atmosphere by the ocean. So I'd say that's more important than any amount of money. So the rainforests of the ocean, they're hotspots of the kind of process that makes life possible on the planet. And they also shelter most juvenile fish on the planet that aren't farmed at some point shelter in a coral reef. And they're threatened by all kinds of stuff, by pollution, by destructive fishing, and particularly by climate change. Ocean warming kills off the microbes that give them the color, and coral bleaching, which looks like this. Acidification dissolves the skeletons of coral reefs and turns them into, unfortunately, piles of rubble. And how we study them is we create these things called arms, which are just stacks of 8 1-foot by 1-foot plastic plates. We bury them to the ocean floor, and when we recover them, about 18 months later, they look like this on the bottom right. All kinds of life has recruited onto this plate. And by studying the biodiversity on this plate, we get an estimate of biodiversity at the reef. Well, how do we study it? The thing is, I worked with NOAA on a project that generated 14,000 of these. And so we can't study them by showing them to people and saying, hey, what's on this thing? We can only do it by showing them to computers and developing computer models. So in the interest of time, I'm not going to go over the big idea of neural networks. But when we combine them with these photos, we have the project that I call Coral Vision. And the big idea is to use deep learning, machine learning to identify what's on these plates, and then to empower the whole global research community by making a public website that will let anybody identify what's on any one of these plates, and then correlate reef health to what's going on in the ocean. So that's what's going on underneath the ocean. I have other projects that are similar. And then there's outer space, because it turns out that bioinformatics is very useful to NASA in solving this horrible problem. The problem is that when you take a human body that spent all these millions of years evolving to live on the surface of the planet, when you put it in low Earth orbit, health is seriously challenged. And so working with NASA, I created the Spartan Space Mines program, where students who take the undergraduate bioinformatics course and minor in bioinformatics can get special training and go to NASA and do research. And also my research group does additional research. Here's an artist rendering of one of the outposts that they're planning to send to the moon as part of NASA's Artemis plan. So just to back up, this logo is the logo of GeneLab, which is a repository of genetic data from literally decades of experiments that have been flown in space shuttles and satellites and the International Space Station. And so we'll have a control animal on Earth just under normal gravity, and we'll have one in microgravity, and we'll measure its genetic response and to see what genes are being expressed. And by seeing the differences between ground control animals and spaceflight animals, we can see how spaceflight is affecting the health of animals who go into outer space. Because for all I know, one of you listening to this may be one of our next animals to go into outer space. And if that should happen, I wish you good health. I'm always looking for talented students with sharp programming chops. And if you're interested, I've put my, let's see, did I put my email address on here? No, I didn't, but I'll bet it's going to show up in the chat any second now.
It is, and we have to make sure it's 1 L, not 2.
Yes, 1 L in my first name.
Yes. All right, thank you very much. We appreciate it, Philip. Next up, Ms. Taylor Dawn Francis.
Hello, everybody. Thank you for this opportunity to allow me to speak to you all about the Beginner Scholar Program and what a wonderful opportunity it is for students to be able to conduct research with mentors. Our whole mission is to diversify the professoriate and graduate schools in that space. We give an opportunity for first generation low income students to be able to see themselves doing research like conducting research and be able to become a professor. So they do that by pairing them with a research faculty mentor in their specific field. As Dr. Journey was speaking earlier, Brittany is one of our students. She's currently in our current cohort. And then Dr. Walcott was speaking earlier. He has been a previous mentor. So our pool of mentors is just only growing. It's great for students to be able to do this, right? They conduct research in the summer. They're also able to participate in summer research programs at other universities. We help them get into them. We also give them a chance to present at research conferences at various schools across the nation. So before all the students, as travel, air travel, right, hotel is all paid for by the program. So the class that we offer, right, is 3 units, spring semester, pass no pass. We teach students how to write a research journal, right, how to write a literature review, create a CV, craft a personal statement, all these things that will help them get into these graduate school spaces. The research aspect of it, we go over in this course. So all the fundamentals of completing research, this is what we do, as well as build community. So the cohort, they're able to work together. It's from all across different majors. So we have STEM, or we have anthropology, we have humanities, right? So they all work together in completing their research projects with their mentors. So the McNair Scholars Journal, students have an opportunity to publish in our journal. I know many undergraduate students don't get to. So the culmination of the spring class and the summer research opportunity that students get to do with their mentors becomes the article that they do in the journal. So we have not only a physical copy, we have also a copy online. So there's the link right there. It's filled with so many amazing works by different students all across. I think it goes down up to back to 2016. So you can just look and see what other students have done with their mentors, what kind of research they've conducted. So the stipend, we have a $3,500 stipend this year. It has never been this amount before. It used to be less. This is contingent upon the student completing other responsibilities for research. They have to participate in the program, finish all of the assignments that we give them, which are slowly build toward them completing their research projects with their mentor. And then we also offer the graduate boot camp to students, which is every June. We host it. We have people from all across the nation come and speak to these students, about the PhD application process. We have GRE test taking tips. Again, we go over the personal statement, CV, letters of rec. We also help students reach out to faculty advisors at these other institutions because they want to work with them. So we want to build a rapport. We want these students to build a rapport with the people that they could potentially be working with. Then we implemented self-care during graduate school just recently, and then experiences from former McNair scholars at San Jose State and other universities, just about them working with their mentors and the experiences they've had in the program. So we also have university tours. We take students to different campuses. I've personally taken them to Harvard and MIT, so students can see themselves in these spaces. It's a really, really great opportunity. And they can learn about these PhD programs and maybe reach out to professors that they want to work with at the university. We've had students correspond with professors over email. And then when we set up the meeting, when we flew out there, they were able to meet with them and see the opportunities that they'll be able to have. So the timeline is we really want students for a whole year. It really takes a good amount of time to cultivate research. Some students come in really green and they have no idea what they're doing. So we kind of work on different spectrums. Other students are really experienced, they hit the ground running and they already have a mentor. So it's just an array of different students we get. So the class happens in the spring 2022, and then research will be conducted in summer 2022. And then the culmination of that will be the McNair Journal in spring 2023. So everything you've been working up toward from spring gets finally published. The fruits of your work. It's published in spring 2023. So there's my contact information. I believe it's going to be put in the chat as well. This is a really great opportunity for students. I've been working with the McNair program for about 6 years. We've seen many students come and they've been able to get into, NYU, Johns Hopkins. It's a great experience, and it's good to see, for first generation low income students be able to get into the spaces that they want to get into. So if anybody has any questions, I'm more than happy to take them.
All right, so thank you very much for all the presentations. Fantastic that everyone got them done under an hour. All I've been thinking of is there a way for me to track a bird in low Earth orbit that typically resides near coral reefs as it rips through space-time, simulated, and have a microfluidic device in its back used to understand clotting mechanisms as the bacteria in its gut recycle rare earth materials and nanodiamonds, and then have this team together work on it and then become a McNair scholar. But, but I'm not sure that would happen. But I mean, please feel free to use the remaining 5 minutes to connect with one another, because I know some of the professors have not connected before. So if you want to ask any questions, if the students want to ask any questions, you could place it in the chat and connect with the professors. We truly appreciate your time. This event came about because, I remember one of the students talk while I was talking about, the students don't typically know about the STEM research opportunities at SJSU. So your presentation is going to be recorded and it will be shared with them, and hopefully some of them reach out to you to take advantage of the opportunities. So thank you very much.